Hi, I'm CG Sigmund, and for this project, I researched autosomal recessive disorders in Hutterite populations. So I wanted to start by explaining a little bit about who the Hutterites are. It's a small subset of the American and Canadian population. They descended from 89 original founders, part of a isolated religious group in a white European area, and they're closely related to the Mennonites and the Amish. So these 89 original founders migrated to America after experiencing religious persecution in Europe and founded 3 small settlements called lutes. Over the course of 10 to 12 generations, through interbreeding and practices like large families, the Hutterites have been able to expand their numbers from 89 to 40,000. Um, because of closed cultural practices such as interbreeding, only marrying other Hutterites, they've really been able to limit gene flow between outside populations. And this means that they are really prone to displaying the founder effect, which I will discuss later. Okay, next I wanted to discuss and define some autosomal recessive disorders. An autosomal recessive disorder is a type of Mendelian disorder caused by 2 homozygous recessive alleles. So the offspring will only display this disorder if both of the inherited genes, one from each parent, is the mutated recessive allele. So one common example of an autosomal recessive disorder that is particularly important for this investigation is cystic fibrosis. This occurs when an offspring receives 2 mutated copies of the cystic fibrosis transmembrane regulatory gene, and it results in an inability to regulate the mucus production of the body due to a deficiency in chloride at the cell surface. So chloride, the type of salt, is not readily available at the cell surface, which doesn't attract enough water to the cells, meaning that mucus buildup in the lungs can affect respiration, it can affect digestion, and it progressively gets worse with age. So the most common mutation that causes cystic fibrosis among white Europeans is the delta F508 mutation. This is carried by about 5% of white European Americans. However, there's an extremely rare mutation, the M1101K gene mutation, that also causes cystic fibrosis that is extremely common in Hutterite populations compared to other white European groups. It is almost unseen outside of Hutterite populations. So this has led researchers to conclude that this mutation must have occurred in the founding generation when recombination of the CFTR regulator led to a mutation in the regions between the 14A and 17B exons. Essentially, cystic fibrosis is a very important autosomal recessive disorder when examining genetic variation between Hutterites and other white Europeans, because although they both display the same phenotype of cystic fibrosis, Hutterite populations have a unique genetic mutation that leads to this phenotype. Some of the other common autosomal recessive disorders that are particularly common in Hutterite populations are congenital deafness, types of muscular dystrophy, and Bowen-Conradi syndrome. However, cystic fibrosis is probably the most important for the sake of this investigation because it allows us to compare variants between populations. Okay, so next I want to define gene flow and founder effect because these are largely the reason for the unique genetic makeup of the Hutterite population. So gene flow occurs when individuals enter or exit a population, and it alters the allele frequency in that population. The founder effect is a type of gene flow in which a small subset of an existing population migrates away from a larger population, forming a geographic and genetic isolate. The founding members of this new population are able to foster new generations, but these new generations have significantly decreased genetic variation as opposed to the larger subset of the population, because the only genes available to these offspring are the ones that were passed on from the founding population. The only source of new alleles would be mutation, and if you are like the Hutterites and limited gene flow outside of founder effect, then it is likely that autosomal recessive disorders and other genetic patterns of inheritance become noticeably marked in the population. Hutterites are a really prime example for modeling some of these disorders because of three different factors. The first is that they were founded by a small known group of individuals, so this makes tracing ancestry much simpler and clearer. Also, their closed monogamous marriage practices, it actually limits the amount of genetic variation that is available to each of the offspring. However, through monogamy, it enables researchers to trace pedigrees, which I'll discuss on the next slide, to determine carriers of genetic disorders. Lastly, the Hutterites experienced a very rapid population expansion in a really restricted period of time, which makes it ripe for younger offspring generations to display mutations of the founders, because there have been very few generations for these mutations to be like completely eliminated from the gene pool. So we're only 10 to 12 generations out from the founding. Therefore, a lot of the offspring still maintain genes from their founders. However, the rapid population expansion means that there's a significant portion of the population that displays these genetic mutations from the founders. Okay, so the last thing I really want to talk about with Hutterites and autosomal recessive disorders would be a tool for mapping their inheritance patterns, pedigrees. So these are a type of diagram that maps an organism's ancestors, and they're especially helpful in determining the carriers of autosomal recessive disorders. If you look to the right here, you'll see an example of a pedigree that maps autosomal recessive disorders. As you can see, the two founders, a male and female, were both heterozygous, which means they're a carrier of the disorder without displaying the disorder. And in the following generation, you can see that with each passing generation, there are fewer offspring that display the disorder. However, there have been so few generations between the current 40,000 Hutterites and the founding 89 that these genetic disorders have they have not been weeded out of the gene pool, and they are still extremely common. So here are my citations for the photos I used in the slides. I really hope this was helpful. If you have any questions about the Hutterite population or autosomal recessive disorders as a whole, you can leave them in the discussion forum and I'd be happy to answer. Hope you enjoyed. Thank you.